Okay, so we hit what looks like to be quite a large chunk of key knowledge, which is just presented to us as nucleic acids and proteins. So to begin with, we'll just look at both of those as macromolecules and their monomers and little key things you can use to recognize them and some basics about them. And then it builds into some more detailed knowledge on how they function, so what we know about proteins. I'll read through it. So nucleic acids and proteins. Nucleic acids as the information molecules that encode instructions for the synthesis of proteins in cells. Protein functional diversity and the nature of the proteome. The functional importance of the 4 hierarchical levels of protein structure, the synthesis of polypeptide chain and amino acid monomers by condensation polymerization, the structure of DNA and the 3 forms of RNA, including similarities and differences in their subunits and their synthesis by condensation polymerization, and the genetic code as a degenerate triplet code and the steps in gene expression, including transcription, RNA processing in eukaryotic cells, and translation. So it is a large chunk that is related to nucleic acid and protein structure, how they're related to each other, and then what they actually do in the cell. All right, so quickly, there's basics about the monomers that they're just need to know facts. So looking at the chemical representation of the monomers, can you name all 4 of these? So hit pause if you need to. I'll just keep talking. You're looking at the chemical structure, which you would never have to be able to draw, although a point of contention with the amino acids, it is worth being able to at least show the NH2, the carbon, the extra hydrogen, the carboxyl group COOH, not necessarily showing these bonds, but being able to write it as COOH, and the R group at the top. You may even get away with writing amino, carboxyl on this side, hydrogen and the R group all forming around the center carbon. That's probably the only one where you do need to be able to draw from scratch a chemical structure, although it's not entirely certain that you would have to do that. But you must at least be able to recognize them. So you should have labeled this one as the amino acid. This one is your nucleotide forming nucleic acid as the polymer. This one is a monosaccharide. Specifically glucose, which is a carbohydrate monomer. And this one here is our, from the last video, if you watched that one beforehand, you would recognize that as being phospholipid structure, or at least a form of lipid. And you can see that the fatty acid tail gives away that as part of its chemical structure. Why have I got both of these crossed out? Because all 4 macromolecules used to be a really strong focus for biology. In the most recent version of the study design, not the 2021, but the one that we are working from anyway, they have been greatly reduced in their importance. So carbohydrates, pretty much as long as you can recognize the hexagonal structure as representing a carbohydrate, and lipids, you certainly wouldn't have to draw out the chemical structure, but they are still important because we focus on the cell membrane, and knowing what a lipid is still of some importance. But by far, this section is focusing on amino acids, polypeptides, and nucleotides forming nucleic acids. So straight off the bat, quick multiple choice question. This one says the most appropriate representation of a triglyceride, but can you rephrase the question? Can you just identify all 4 of these? So hit pause, answer, and straight away the triglyceride would be this one. So this is representing the glycerol head and the 3 fatty acid tails, but just being able to recognize that that is a lipid structure is enough. What are the other three then? So again, hexagons. Hexagons trigger in your mind, hopefully, monosaccharides, and therefore when they're all joined together, this would be a polysaccharide or a carbohydrate. This one is probably easier to recognize. It's got a more unique structure. Instead of having the hexagon carbohydrate as part of its structure, it's got a pentagon. And so a pentose sugar attached to some things that are joining together in little circles. Hopefully you looked at this and you went, yeah, that's nucleic acid, and specifically this would be DNA. How do I know it's DNA and not RNA, which we'll harp on a little bit more about later? It's double-stranded, so that one is DNA. And then at the top here, so for answer B, we have a circle joined to a square, joined to a star, joined to a hexagon. And that's just meant to represent that we've got different monomers that are different to each other coming together to form a polymer. And that should scream to you amino acids forming a polypeptide. All right, so we'll look at our proteins first. Okay, so basics for proteins, things that you must know. This is a good summary slide for you to just pause on and see if you can elaborate on these points. So this would be the chemical structure. Like I've already said, being able to draw this chemical structure, bit of a grey area as to whether you need to be able to do that, but at least have the basic layout down. So this is the amino group, the carboxyl group, another hydrogen attaches to the bottom of that central carbon, and the R group at the top here. So just, I know it's a little bit further down the list, but to talk about it while we're looking at the chemical structure, R is not an element, so why is it being represented here in a chemical structure? Can you answer before I tell you? It is the variable region of the amino acids. So we know amino acids have a huge variety. There's 20 different amino acids. They all have this constant structure here, but they will each have a different R group. So from the smallest amino acid, which has just another hydrogen attached here, to a very large amino acid, which might have extra ring groups hanging off it. But they all vary in this R group. And that's what, in the end, allows a protein to take on a unique shape. It is the interactions between these R groups that will dictate how a protein folds. So if you have a negative and a positive R group sitting at length in a polypeptide chain, they might want to sit near each other because the whole opposites attract thing. So we'll bring them closer together. Different R groups have different affiliations for other amino acids, and so that's what brings it together and gives a protein its folding shape. Okay, so the monomer is amino acid. The polymer is polypeptide, not protein. So not every polypeptide is a protein. So that is a good distinction to make when you're defining these terms. They are held together by a peptide bond, and so all three of these words should be something that you are associating with proteins. Levels of structure. Before I run through them, can you just jot down what you know about these levels of structure? What is primary, what is secondary, what is tertiary, and what is quaternary? So there are key words that you should have sort of in mind for each of those. Okay, so hit pause and you define. Primary, hopefully you said, is the order of amino acids in the polypeptide sequence. So which amino acid goes in which order. Secondary, it's these structures that you can see in this three-dimensional diagram here. So I can see these coiled up structures. That would be an alpha helix, and I can't see any of this diagram, but the flat arrow structure that we've seen in a previous one would be the beta-pleated sheets. So when the protein, when the polypeptide folds to form alpha helices and beta-pleated sheets. If you're asked to define secondary structure, as long as you say those two formations, you have the mark. Tertiary structure, further three-dimensional folding based on interactions between R groups. for a polypeptide chain, which brings us to quaternary. The key word that you should have in your quaternary definition is more than one polypeptide chain will join together, and that is in a diagram represented usually with colors. So we can see two blue polypeptides that are separate to two red polypeptide chains. They would make it blindingly obvious to you in an exam question that you would have a different shading, a different coloration to those polypeptide chains, or the question would state that it's made from multiple polypeptide chains if you were being able to identify or being asked to identify quaternary structure. We've already talked about R groups and their significance. The proteome. So this was a question, I think, in our trial exam. What is a proteome? I think people are very familiar with the word genome, which is all of the genes that are found within a species. The proteome is all of the proteins that are found within that species. So the set of functional proteins found within a species is its proteome. Proteins can be fibrous or globular. So these are two words that are sometimes used to describe proteins in an exam and can really throw people. So just a reminder, globular would be something like this, where the protein has folded into a sort of shortened, three-dimensionally shaped glob. Whereas fibrous, they are more structural proteins. So something like keratin or the proteins that make up the cytoskeleton in a cell, they are elongated, drawn out, long proteins that are used for structure. So that is fibrous versus globular. And of course, we did lots of work at the start of the year on enzymes, and that was your extended investigation where you were looking at the function of catalase. And so everything that applies to proteins would be used to describe an enzyme, because enzymes are proteins. So their structure is the same, just we talk more about their structure having active site, and words like substrate get used, and we talk about optimal conditions for enzymes. But don't forget, enzymes are proteins. So if you're asked about the structure of an enzyme, it is the structure of a protein. So worth noting. Okay, so the condensation reaction. You can be asked to describe the condensation reaction, which is, they are essentially asking you to describe how two monomers of these macromolecule polymers join together. So the example that I've used here, and that I would have used when we went through it at the time, is amino acids. So two amino acids coming together to form a polypeptide. The same reaction happens though when two monosaccharides are joining to form a polysaccharide, or when two nucleotides are forming a nucleic acid along that sugar phosphate backbone. The same reaction will happen. Or when the fatty acid tails are joining to the glycerol head for a lipid. Okay, so we're using amino acids as our structural representation, but the reaction is the same no matter what the monomer and the macromolecule, the monomer and the polymer. So condensation means that a reaction is going to occur between these two monomers that produces water. That's where the name comes from, condensation, water forming on your windowsill, water formation in chemistry as well. Condense, I guess you can remember it as squish together. They're coming together. And they are anabolic. And that little reminder, that little word that you can use to remember those three words going together. So anytime you're talking about condensation or building a polymer from two monomers, it is an anabolic reaction, it is condensation, and it is endergonic. You are building energy into that newly formed bond. And it's where monomers are joining or subunits are joining together. So you can see here, going from that side to this one, the hydroxyl, the OH group from this side is joining with the hydrogen from this side. And when those two come together to form water, it allows these two, the nitrogen and the carbon, they've got a free bond to make then, and they can make that now with each other to form the peptide bond between the two amino acids. So we only call it a peptide bond if the monomers we're talking about are amino acids. The opposite of that reaction would be hydrolysis. So that is where we are now using water to split the monomers. And so the other word that we can associate with these reactions is CHEX. So ACE for building, CHEX for breaking. It is catabolic, C for catabolic, H for hydrolysis, and EX, exergonic. So we are breaking it apart, reforming those bonds with the hydroxyl and the hydrogen, and we've now got our two separate monomers again. So all of our macromolecules can be built together or they can be broken down depending on what we need to happen with them. Obviously, the breaking down is a bit of a hierarchy. So your body will go for carbohydrates first to release energy from. If no carbohydrates are available, then your body will use fatty acid stores. So we'd do this reaction, breaking down our fatty acids. In a process of starvation, then your body might start breaking down your polypeptides, your proteins, and actually releasing the energy from that peptide bond in order to use as useful energy in your body. And nucleic acids could eventually be an energy source as well. So all of your macromolecules in your body are a potential energy source using this hydrolysis reaction. Okay, looking at nucleotides now. So this is our nucleotide structure. You don't have to draw the chemical structure of a nucleotide, but you would be called upon to draw a simplified version of that. So being able to show the phosphate attached to that pentose sugar, which if it's RNA, it's a ribose sugar. If it's DNA, it's a deoxyribose sugar, and attached to a nitrogenous base. So that three-part structure to the nucleotide. Differences between our DNA and our RNA. Hopefully these spring to mind pretty easily. I think I've got them listed, which is fine. We can just talk about them. Okay, so DNA, when we're talking DNA, we know it's a genetic code. Where do we find it in a cell? It's located in the nucleus, in the mitochondria, and in the chloroplasts of a cell. And remember, the fact that we find DNA in mitochondria and chloroplasts is evidence for that endosymbiotic theory that those two organelles were their own organism before they were engulfed by the developing eukaryotic cell. RNA, what forms do we find RNA in? Messenger RNA, which we talk about in transcription. tRNA, which comes about when we're talking about translation. And rRNA, which we never really discuss in great detail, but we know that it's part of the structure of a ribosome. Differences. How can we tell that a diagram is showing us DNA or RNA? Can you list off those differences? I think we should be able to come up with 3 things that we're looking for to tell the difference. Okay, so it is double-stranded versus single-stranded first. So double-stranded DNA, single-stranded RNA. Your pentose sugar might be labeled as being ribose or deoxyribose. So here it's labeled as deoxyribose, and I can see that the diagram is showing me a double-stranded molecule. So I will say that this is DNA. And that base difference. So for DNA, we have thymine as one of our bases. For RNA, we have uracil as a base. So just like the proteome was the set of functional proteins for a species, here we're talking about genomes. Genomes would be all of the genes of a species, which have been a lot of work's been put into mapping these for the Human Genome Project, and we now know that there's roughly 25,000 human genes, and they're loci where they are can be mapped to a position in our karyotype on a chromosome. Definition of a gene. If we're going to talk about gene expression, then we need to remember what a gene is. So a section of DNA that codes for a polypeptide is the short and sweet definition. Roughly 25,000 genes in the human genome, which can range from between 100 to 2.5 million base pairs, so they can be very small or very large. That the genetic code is that sequence of A, T, C, and G that we've become familiar with, and that's read in triplet that we call codons. So each gene is read in sets of 3 bases at a time, and that set of 3 bases will correspond to an amino acid to build the resulting protein or polypeptide. Not all DNA is part of a gene, so there's a lot of in-between DNA, some of it's still being researched to discover what it actually does. But a lot of it is involved in regulation of genes, so it might not be the coding region itself, but regulating that gene activity. And there is some junk DNA, even within a gene, that we call introns. Okay, gene expression. So when we're talking about gene expression, we need to start with transcription. So this is where we are recognizing that RNA polymerase is an enzyme that will bind to the promoter region of a gene. So P for polymerase, P for promoter. And it will move along the DNA and transcribe the template sequence. And transcription involves the RNA polymerase attaching the complementary RNA bases to the DNA template. So when we read A on a DNA template, it is pairing with U becomes the biggest factor to remember. So hopefully that looks familiar as a process. Don't forget that RNA polymerase is the enzyme involved. Okay, so I don't have a slide for RNA processing, so we just need to talk about it. Before this RNA can be called mRNA, we would actually call it pre-mRNA right now, because there's 3 things that need to happen to it. I might skip back a slide to talk about it. Because each gene is made up of regions called exons and introns. So exons are parts of the mRNA after it's transcribed that we want to keep. They are expressed. Introns are in between sort of junk DNA, so bits that we want to get rid of. So RNA processing involves 3 steps. Can you name all 3? Okay, so hopefully you came up with splicing, which is what I just described as the removal of introns, and exons are being kept. They get spliced together to form the end resulting mRNA. The addition of the 5' cap and the addition of the poly A tail. So those 3 steps for RNA processing. If you've got a 3-mark question about RNA processing, they're the 3 things that they want you to say. And then we have our finished mRNA at the end of that processing sequence. Once the RNA has been processed, it is free then to leave the nucleus and travel to a ribosome. As I mentioned before, we might have a ribosome that is free-floating in cytosol and a ribosome that is attached to the rough endoplasmic reticulum. It depends on what kind of protein we're making. If we're making a protein that is going to be used by the cell that's creating it, then it's going to go to a free-floating ribosome and the protein will stay within the cell. If the protein is destined to be exported from the cell, then it's going to go to the rough endoplasmic reticulum to be made and folded, and will then be able to leave the cell. So it occurs at ribosomes, rough endoplasmic reticulum or cytoplasmic, as I've just described. mRNA is read in triplet codons. The start sequence is always AUG, so the mRNA will move through the ribosome until it hits the sequence AUG. And then the first tRNA will add on that first amino acid, which is always MET, because that's the corresponding amino acid to the AUG. So we can see here a diagram showing, I might just move the screen down a tiny bit if I can. Ooh, no, I can't. There we go. Right, showing how our mRNA, we've got our codon on our mRNA, GAG. We would look up on the universal code, what does GAG code for? And it should be, well, the amino acid's not shown here, so it should be the amino acid that's attached here. The way the tRNA doesn't carry around a universal code to look up, the way the tRNA recognizes that its codon has come up is because it has an anticodon as part of its structure. So for the codon GAG, the tRNA that has the anticodon CUC would be able to bind in the ribosome and it would drop off its amino acid. You can see a different diagram up here sort of showing that process. The codon says UCG, the anticodon says AGC, so it can attach and drop off the next amino acid, which will be SER here. Okay, so 20 different amino acids can be combined in a particular sequence, and it's specific because every codon specifically codes for one amino acid that will be added in order. And if we get the correct order of amino acids in our primary sequence, that allows the folding to take place normally and we'll get the correct shape to our protein. All right, so some practice questions. There's quite a number of practice questions here, so I will try to just allow you to pause and then I'll run through the answer and we'll skip to the next one. All right, so when the next question flashes up, just hit pause straight away, answer the question without me describing anything, and then you can check your understanding as I run through them. Okay, so a diagram is represented here, and hopefully you can quickly recognize it as being DNA. There is some sort of unnamed molecule that is producing this long structure after it's moved along this DNA. The diagram is most likely to be of definitely transcription, so B for that one. The molecule labeled X represents, and hopefully you said RNA polymerase because we know that that is the enzyme that's involved in transcription. Okay, hit pause and have a read. Okay, so our strawberry genome is found in all cells of the strawberry that contain a nucleus, so that one's false. It includes all of the proteins, that would be if the word said proteome, comprises all the genes, definitely that is our definition for genome, so C for that one. It is expected that an offspring produced from sexual reproduction of F. vesca does not translate all of its 206 million base pairs because we know that not all DNA is coding region. Adenine and cytosine are not base pairs, so that one is false. Does not have to have equal numbers of those two. And that is a common question that gets asked to work out the amount of one or another base based on how much of one of them is. So you would always have to remember that adenine will pair with thymine, cytosine pairs with guanine to work out the maths of that. Receives half of its chromosomes from its female parent. Basics of sexual reproduction, you are receiving half of genetic information from each parent. And the same combination as other strawberry plants from the same parents. No, that's effectively saying that you are this identical copy of your siblings, which we know is false. Okay, so next question. Now I have to move the screen around a little bit for this one, so have a read of that first one and then hit pause. Okay, so the DNA template strand can code for serine with any of these codons, which a tRNA molecule that codes for the amino acid serine is. You can immediately eliminate A and C. Can you see why? Because if it's tRNA, it's not going to have a T in it, because RNA will not have thymine, it will only have uracil. So it's either B or D. And of B and D, you're looking for one that would be able to pair up with this DNA. So there's a little bit of a trick to the question. Because it's a DNA template, you need to take into account the fact that it would be transcribed first to mRNA, and then the mRNA would be what pairs up with the tRNA. So we are looking for, say D, UGC would have to pair with AGC, which in the DNA would pair with TCG. So TCG is one of these sequences, so your answer should be D. I'll move the screen down a little bit so you can see the rest of question 12, and if you hit pause. All right, so this one takes a little bit of reading and understanding, but hopefully you come up with an answer. Okay, so the HOC gene on this plasmid codes for a toxic protein that causes cell death, which is represented here. And the SOC gene, if transcribed, can intercept that pathway and lead to breakdown of the mRNA instead of the toxic protein. So this is a nice example of a diagram that, again, you're unfamiliar with. It's not something that we have studied, but you have the equipment, you have the skills to be able to digest the information and come up with an answer. So it would be reasonable to expect that HOC mRNA would be translated if SOC mRNA was absent. So if there's no SOC mRNA, for this to happen, that the mRNA will be translated. So that requires you to know what happens after this, between this step and this step, that after transcription, translation needs to happen to make the protein. And that's true. So that is what the diagram is showing us. So your answer would be A. B, HOC DNA would be unable to be transcribed if SOC was present. So if SOC is present, the RNA is still made, so it's still transcribed. So you need to be able to recognize that that is transcription happening there. So B would be false. SOC mRNA would have the same nucleotide sequence as HOC. If they are joining together, they are not the same sequence, but they are complementary sequences. So a little trick to the wording. An E. coli cell without any plasmids would be killed by the toxic protein. If the E. coli cell did not have the plasmid, they would not produce the toxic protein because the gene for the toxic protein is on the plasmid. So your answer would have been A. All right, have a go at these ones and hit pause. All right, so question 2, triglycerides, lipids would have been B. They are a rich source of energy. I think it's a common thing for students to forget about lipids being a source of energy. It is accurate to claim that lipids are packaged in mitochondria. No. Do you know the organelle that is responsible for lipid modification? Smooth endoplasmic reticulum. Yes. Carbohydrates increase the rates of reaction. So it's talking about an enzyme there, and an enzyme is not a carbohydrate, it is a polypeptide. Nucleic acids are made up of chains of amino acids. Nucleic acids are made from nucleotides, so no again, which leaves us with D, which is hopefully right. Polysaccharides can be found as starch in plants. Good going, yes. Starch is a very good example of a large polysaccharide found in plants. A molecule of transfer RNA could include the nucleotide sequence. Did you see the trick in this question? It was actually turned out to be a really quick question because there was only one of these sequences that did not contain T-thymine as part of its sequence, which would not be found in RNA. So B had to be your answer. And 6, list the 3 components of a DNA nucleotide. Phosphate, DNA nucleotide, so you've got to be specific, a deoxyribose sugar, and a nitrogenous base. I have to move the screen again, so bear with me. Okay, have a go. It was a bit of a tricky marking scheme for this first question from memory. You'd think 2 marks for saying that Sally is correct and why she's correct, but instead go for a comparative statement. So Sally is correct. It is cellulose, and state a reason why it's cellulose and a reason why it's not cellulase. So cellulose is a carbohydrate, and this is a carbohydrate because it consists of the elements carbon, hydrogen, oxygen. That would do. Whereas cellulase would contain the element nitrogen and be composed of amino acids. That would be 2 marks worth. Okay, the question down the bottom, have a read and press pause. Okay, so again, another question where you're going to read it and go TLR, toll-like receptors. I don't remember learning about these. I don't remember reading these. And it might throw you off your game, but all you're being asked is the name given to the type of secondary structure that is labeled W, and hopefully alpha helix is the term that you wrote down. All right. So behind my picture will say GC receptor, so I'll just cover that up. Hit pause and have a read. Okay, last question for this section. There is a larger question that goes with this. It's about hormones and signal transduction, but the simple start to the question is asking, name the enzyme that catalyzes transcription. And being fresh in your memory, we would know that the enzyme that is catalyzing transcription is RNA polymerase. Name the transcription product, molecule S, up here. And describe the processing that molecule S undergoes before it exits the nucleus. So we know that this is pre-mRNA because we're now referring to RNA processing. So having a look at the marking scheme for this question. Back on. So naming it as pre-mRNA and then describing 2 of the processing events. I think they accepted another answer because the question might have been a bit ambiguous. But what they were really looking for in the first place was introns removed and exons joined together, which is your explanation for splicing. The addition of a 5' cap, which is the same as saying the methyl cap, and the addition of the poly A tail. So that's a good example of a set of answers that they're looking for RNA processing.